Hi, hello and welcome to the Illuminating Immunity Virtual Summit, Light, Photobiomodulation versus the COVID-19. I have as my guest Professor Janis Eells. She is a PhD in pharmacology and toxicology from the University of Iowa in 1981. She's also a distinguished professor of the University of Wisconsin Systems. She's a pioneer in photobiomodulation. She's been ahead of the game for quite some time. She was the very first researcher to look at the retina, which is a completely wonderful application that she's come. And so we're very happy to have her as a part of the summit. Thank you, Janis.
You're welcome. Thank you.
So you were very quick to turn your attention to light and the COVID thing. You're one of the first persons that started this. How did you, everything was clicking in your mind. Tell me the story a little bit. How did you start producing this perspective?
Now, are we talking about the perspective for this symposium, or are we talking about the symposium? Okay, for this one, one of the things that has become evident in retina research and actually in any neurodegeneration research is the role of the dysregulation of the immune system and the role of inflammation. And one of the characteristic features of photobiomodulation and characteristic treatment advantages is that in general, it is very anti-inflammatory. And it modulates the immune system and immune function in a way that is generally therapeutically positive. One of the reasons I jumped in here is in some ways to insert a little caution in moving forward, because it's not always positive. And there are cases where, and I'm going to cover those, where stimulating the immune response can actually cause it to run amok, which already happens with COVID-19. And in fact, we don't really understand the pathogenesis completely of the disease. And so what I wanted to do in this talk was basically, and a lot of this I've been learning on the fly very quickly. A lot of this is very new to me. I am not an immunologist. That would be Dr. Jerry Lyons. But Dr. Lyons and I have collaborated for a long time, and we have actually observed situations in which treatment with photobiomodulation, especially where we used red light, was harmful to the disease process. So what I want everybody to do is sort of stand back and take a deep breath before you run full force at shining lights on people with disease.
I would agree with you, and I think that's been brought up throughout the summit. But remember, we have a gas effect and we have a brake effect on the immune system, just to put it kind of generally. And so right. It's kind of the orchestration of those, which we really haven't. Maybe we don't have enough clinical data. So all of these observations and putting the research together is going to help us all.
Right. I totally agree.
So let's go shall we go to the slides?
Let's go ahead and do it.
We have illuminating immunity, COVID-19 induced immune dysfunction, the therapeutic role of photobiomodulation.
All right. Thank you very much. I would like to focus my talk on looking at the immune dysfunction as we understand it right now with COVID-19, and this is a ever-evolving process, very rapidly evolving, and look at what we know about photobiomodulation and what we know about photobiomodulation and viral sepsis, which is the closest model we have to COVID-19, and then draw some conclusions. So what I have submitted, and this is sort of my overall thing, we can jump to the bottom line here, is what I want to talk about is I want to go through the pathogenesis and the induction of sepsis by SARS-CoV-2 and talk about the potential of photobiomodulation, and then add some possible caveats to whether or not how we want to move forward, because we want to move forward carefully. So I've been in this field for over 20 years now. Photobiomodulation is older than my involvement in it. It's not older than me. But as a science, in terms of low-level light therapy or photobiomodulation, probably applied for over 50 years. Certainly light therapy has been with it since ancient times. And one of the initial individuals who really moved the field forward was Tina Carew, who's the head of the Laboratory of Laser Biology and Medicine in Russia, and she really established the field in terms of the medical uses of lasers, as well as the hypothesis, one of the mode of actions of far red to near infrared light, activating the cytochromes cytochrome oxidase in the electron transport chain. Andre Meister sort of picked up the baton from there and started working when we had laser, looking at, actually he was, I think, preceded that, looked at laser-induced hair growth. Actually what he looked at was he was trying to see if ruby laser, red laser, would either reduce or would eliminate cancer or would exacerbate it. It turns out that in his mouse model it did neither, but it did cause their hair to grow back faster. So, and that actually turns out to be an application that's sort of moved forward. So I've been thinking about light for a long time now. And essentially photobiomodulation, as most of you probably know, is a process by which a chain of biochemical reactions is triggered by exposure to light. And this is everywhere. The way it has to work, photons, light waves, light particles have to be absorbed by a chromophore or photoacceptor molecule. Now take your pick. If you happen to be a chloroplast or a plant cell, your photoacceptor molecule is chlorophyll, and you grow because of that. So these photoacceptor molecules interact with light. Light stimulates a biochemical reaction series in them, and then there is a biological response afterwards. Vitamin D activation, rhodopsin. If you're looking at this slide, light has stimulated the rhodopsin molecule in your photoreceptor cells and initiated a cascade that results in vision. And for far red to near infrared light, another photoacceptor molecule is cytochrome C oxidase. This is my sort of overall pathway hypothesis for the action of light. So you can see the light-emitting diode array here. These are some of the first arrays I used in our studies. Stimulates, we know that it stimulates cytochrome C oxidase. We know that it changes the redox state of the mitochondrion. We know that, and we also know that this only works effectively in disease states, so that if you're treating a normal human being or a normal cell, you get a very mild to no effect at all. But under a disease state, the mitochondria are not working well. Their membrane potential is disrupted. It's depressed. They are producing less ATP. They are spewing out reactive oxygen species right and left and poisoning the cell. The light stimulation turns on cytochrome oxidase, kicks off possibly a nitric oxide that's bound to cytochrome oxidase, basically to try to rein in the dysfunctional electron transport chain. And then the mitochondrion actually at that point can make more ATP. This is a fairly transient effect. In our studies, it lasts maybe 8-12 hours. The longer lasting effects occur because of whatever is kicked off this molecule, nitric oxide, superoxide, hydrogen peroxide, activates transcription factors, and transcription factors turn on or off gene transcription. That's their job. And Michael Hamblin has done an incredible amount of work, and Praveen Arnri has done a lot of work in this area. We know a lot about NF-kappa B and a number of different transcription factors. Gene transcription is then altered, and that alters messenger RNA, which then alters protein synthesis. And I leave this as a simple diagram that altered protein synthesis creates a more stable microenvironment. and in the case we'll talk about today, mostly a microenvironment in which the anti-inflammatory mediators are increased and the pro-inflammatory mediators are decreased. Why is this important? Because this has been widely used in acute and chronic disease states. We know that photobiomodulation exerts anti-inflammatory and immune modulatory effects. I have colleagues who have studied this in heart disease. Our lab studied it in wound healing. We've studied it in neurodegenerative diseases. Other colleagues have studied it in skeletal muscle, aging and injury. And my primary focus has been in retinal diseases. But today I'm going to stretch a little bit outside of my comfort zone into immune modulation. So what do we know about COVID-19? Well, there aren't any vaccines yet, and there are no really well-established therapeutic agents against the virus. There's a lot of testing going on, but a lot of us are still in stay-at-home-to-be-safe mode. The current clinical management includes infection prevention and the control measures and supportive care. There is a focus on repurposed drugs because you can get them out there faster with appropriate pharmacological effects and therapeutic efficacies, including antiviral agents. Hydroxychloroquine has been pretty much tossed out the window for right now in chloroquine, but remdesivir is definitely there, some of the other antivirals. And there's a lot of focus on high levels of ascorbic acid, corticosteroids, nitric oxide, IL-6 antagonists, and in fact, actually, if you look at the clinical trials site on the NIH site, the number of trials for different things for COVID-19 are staggering. So what I did to really put this together is dug into some very, very good reviews so that I could make sense of it, so that I could help you make sense of it. So in order to understand what's going on, we have to have an understanding of the immunological and physiological processes that actually underlie the pathology of the infection. And so I want to go over a little bit of the pathophysiology of the COVID-2 infection of COVID-19, the interaction of the SARS-CoV-2 virus with the immune system, subsequent contribution to this dysfunctional immune response and disease progression, and then, what can we do about that? How can we treat that? What can we do in terms of immunoregulation? So essentially, this is an excellent review that came out probably just a few weeks ago that basically shows that essentially with virus replication and the virus is taken up, it binds to the ACE2 inhibitor and the ACE2 receptor and is taken into the cell, and then the virus just sort of takes over. It starts controlling everything, makes more of itself, matures, and eliminates, and makes more of itself and releases itself. Now what happens is as the cells respond, you get the immune response, you start seeing the interleukins and the T cells and B cells and the entire immune response responding to this. And in the healthy case, on the green side, so follow the green path down on the far left, or the far right, sorry, the alveolar macrophages clean up the neutralized virus, the antibodies get in there, you basically can take care of the apoptotic cells and get rid of them without necrosis and damage to adjacent cells. CD4 T cells mediate an efficient immune response, and the CD8 T cell recognizes and eliminates the infected cells. So the infected cells are rapidly cleared, the virus is inactivated by neutralizing antibodies, minimal inflammation, minimal lung damage. This is actually what happens in most cases. But in some cases, what happens is and then sort of moving over to the left hand here, you start seeing excessive inflammation and infiltration of monocytes, and your immune system turns against you. So there's a systemic cytokine storm, and pulmonary edema, pneumonia, inflammation, multi-organ damage, frequently resulting in death. So this is, we really want to promote the healthy end of it, or find a way to get in before the damage has been done in terms of the dysfunctional immune response. Potential therapeutic approaches so far, antibodies to the spike. So if you can grab that little spike protein that's going to bind to the ACE receptor, before it gets there, you can stop the virus in its tracks. Alternatively, you can look at, because this requires a protease inhibitor can block and prevent the TMPRSS2, and I can't keep track of all the names, but essentially this activates the spike protein through protease cleavage. If you can stop that, you can stop the virus at the cell surface. Alternatively, if you have a virus-specific CD8 T cells from vaccinations, which we're waiting for, you get the granules to actually kill the infected cell. And there's some work going on right now, a fairly novel treatment that targets the cytokine storm symptoms, whereas the blood from the patients with the disease can be passed through customized columns that are designed to trap the pro-inflammatory cytokines before the purified blood is passed back. Now, this is just in the experimental stages right now, but it's a kind of interesting approach. As I said, the numbers of trials are, just unbelievable. This is the first time in my scientific career that I have seen an entire world focused on one question: how do we stop this thing? And so you actually have the kind of collaboration in the sciences that I have never seen, and I hope we can continue. So we're looking at, in China, there's a lot of work that's going on in China, and the Chinese have really been at the forefront of this. Much of what I've read has come from the Chinese scientists. They have been looking at a number of different corticosteroids. So can you, essentially with the cytokine storm, what you want to do is turn that sucker off. Can you, modulate the receptors? What about thalidomide? Thalidomide actually is a great anti-inflammatory agent. So that with steroids, possibly. A number of, there's a bunch of different ones. I'm not going to go through every single one, but there's a whole series of trials going on. There's a fairly new paper, series of papers that have come out that suggest that many of the really critically ill patients develop manifestations that are essentially viral sepsis. They're shock, cold extremities, weak peripheral pulses, even in the absence of actual hypotension. So something's going on with the heart here, and understanding this mechanism is really important in exploring care for these patients. And so there's been some work that has looked at this, looking at autopsy studies and basic science research, and these individuals have hypothesized that viral sepsis is actually crucial to this particular mechanism of COVID-19. And like they say, even though this is all a hypothesis at this point, it's worth moving forward in terms of the research. So viral sepsis basically suggests the normal immune response. The virus is phagocytized. There's an appropriate response of the immune system that the virus is cleared. In immune response in viral sepsis, what happens is there's an inappropriate response. There's viral replication, and there's immune exhaustion. So there seem to be two things going on in this paper, which I'll hit at the very end. just came out that suggests that, that indicates and shows some evidence that there's a two-pronged response. You not only have this cytokine storm, but at some point, the immune system, parts of the immune response have actually become so dysregulated, they're actually turned off. So this, the immune response, this viral sepsis hypothesis is based on evidence that these pro-inflammatory cytokines and chemokines are elevated, and that the cytokine storm plays this important role. And so it's important to identify the source of the storm in response to the infection, to look at cytokine activation, because that's really critical. When does the immune system start pumping out these inflammatory cytokines? Whether or not this is direct virus-induced tissue damage, or is it the systemic cytokine storm, or, as I think is becoming more and more apparent from what we're learning about this, it's the synergistic effects of both is responsible for the multi-organ dysfunction. So essentially, the virus is doing its share. The immune system has turned against these people because they're when they're very, very sick, and this cytokine storm is attacking, and the immune system is essentially attacking the lungs and the other tissues. And is it possible to block some of these pro-inflammatory mediators and affect the clinical outcome in a positive way? And so, and the other part is that not only do you have this stimulation, but there's an immune suppression following this pro-inflammatory phase. And so under this condition, this lymphopenia, so the number of lymphocytes has gone down. This has also been seen in influenza. It's also been seen in other respiratory viral infections. It's been seen in other coronavirus infections. And the degree of this lymphopenia, this loss of lymphocytes, has been shown to correlate with the severity of the disease, but we don't really understand the mechanism. So they have started looking at this. The hypothesis with this study is that in addition to the activation-induced cell death, as well as the tumor necrosis factor-induced apoptosis, that the SARS-CoV is directly infecting the lymphocytes, especially the T cells, and causing them to die. So you've got, so this virus sort of goes at you from all sides. And this dysregulated immune response is shown in this figure here, where you basically have the lymph nodes, the antigen presenting cells, and so you have on the left-hand side of the panel a mild infection where the effector T cells and the macrophages are working, everybody's working fine. On the right-hand, so you can see that particular lung, so here's our lung at the top here, circled gently. This one is gonna do okay. This guy's in a world of hurt. So with a severe infection, the virus invades. The virus can now invade the T cells. It can invade the lymphocytes. It can start killing them off. The cytokine storm starts. You get activated platelets and the fibrin mesh. And one of the problems that I didn't go into here, but is widely discussed, is actually the disseminated intravascular coagulation that occurs in some people as well. And so you get
or to put it mildly, the thrombosis and the thickening of the blood, the viscosity, which is a new development, but we covered it in one of the topics. So
great, good, because yeah, like I say, it's going at us from all sides. So the heart goes, the kidneys, the liver. I mean, there is no organ that is spared in this disease when it is really severe. So here's where, you know, I jump in terms of a real hopeful approach with, and I'm gonna focus on red light, on the 660, 670 far red photobiomodulation No reason to think that near-infrared 800, 830 wouldn't have an effect as well, but these are the studies that have been done. A very good paper, 2017 in Scientific Reports. Essentially, and I'm going to actually walk you through their data because I don't have my own data to show you with this, but I've been through this paper several times. What they did was they induced it's a model of viral sepsis, and I get that. I've been thinking about trying to do some of this work in my laboratory. I'm not playing with viruses at all. We don't even have a laboratory in which we could do that. But lipopolysaccharide, LPS, induces a very nice response, toxic response in the lung, an acute lung response that looks very, very much pathologically and biochemically like viral sepsis. And so what they did was they injected these animals with this LPS or saline, and then they treated them at 2 and 6 hours after the LPS injection. So this was an IP injection. And they treated the animal, and here's their light. I would probably shine light over the whole animal with my LED array at 660 nanometers at a dose of 5 joules per square centimeter. This sort of falls into what almost every study that I've been involved in and seen is what I tend to call the sweet spot for treatment. So photobiomodulation has a biphasic dose response curve. So if you stimulate at 0.01 joule, if you give just a teeny bit of light, if you stand in front of the red exit sign at the door, that's never going to be enough light to treat you. If you give too much light, if you give 100 joules per square centimeter, or maybe even 50, what's going to happen is you're on the other end of that biphasic response curve. It's it just goes up and comes right back down. And that is probably not going to work. Interestingly, it generally doesn't cause harm. It just doesn't work. But if you're between usually 3 and 9 or so, depending on the system, maybe 3 and 15, 20 joules per square centimeter, we usually stick around 4 to 5. We're in that sweet spot that works. It works in the eye, it works for wound healing, and apparently it also works for this particular model of pulmonary sepsis. And so then they administered their light 2 and 6 hours, and then 24 hours later, they did their outcome measures. And what they saw, I'll summarize it here, and then I'll go through the actual figures: a reduction in neutrophils, decreased inflammatory cytokines, an attenuated oxidative burst. And their conclusion, and this is only one of like 4 or 5 different papers that they've done, but I think this was the most clearly presented, is that they find evidence that that photobiomodulation is an effective non-invasive treatment for sepsis. So what they show here is, so they do bronchial alveolar lavage. So you run fluid into the lung, you collect that stuff, and you measure what's in it. And what they showed here was that what you can see is, untreated, LED treated. So these are the guys who didn't get LPS, didn't get the lung toxicity. And then, our sepsis model, the leukocytes just go sky high. They go from, essentially 5 to 30 times 10 to the 4th per mil. And the light treatment, the photobiomodulation, brings them right back down to control, which we see in other things that we look at in retinal disease. It's remarkable how well it works. The same thing with myeloperoxidase. That's a measure of the stress that's going on, of the oxidative stress and damage. LPS skyrockets that, and the light treatment knocks it right back down to control levels. It decreases the granulocytes in the blood without altering those in the bone marrow in the models. I'm not sure I follow all the bone marrow implications, so I'm not going to get into that. I'm actually going to move past this one faster. These are the ones that I like. This is the measurements, and they did a lot of different cytokines in this lavage fluid. And what you can see, again, focus on the black bar, LPS, JAKs, IL-6, or IL-1B up, an inflammatory cytokine. Photobiomodulation knocks it back to control. TNF-alpha, goes from 100 to 200, doubles, knocks it back to control. IL-10 stays the same. It's generally an anti-inflammatory cytokine, so you're not seeing too much effect there. IL-6, again, not quite as much of an effect, but IL-17A is jacked up with the LPS and brought back down to control. So again, the anti-inflammatory cytokines are increased and the pro-inflammatory cytokines are decreased, and we see that in some of the wound studies that we've measured, in chronic healing.
Janis,
Yeah.
Go back one slide there, because
Sure.
you left out the last one in the lower right-hand corner, the IFN gamma.
Oh, right, interferon gamma. Right. So that's actually beneficial, right? And so what you're seeing is that, so there's a response, there's a response of the body to the LPS to ask, let's turn on some interferons. And the interferons, actually, this is what the Cubans have been using in their treatments. They have an interferon treatment that they've had out for viral infections for years. And what happens is, I'm sorry I missed that, is the photobiomodulation actually moves that up even further, so that the body is trying to protect itself, and the light is in fact enhancing that response. Thank you. Sorry I missed that. And then it also decreases the oxidative burst to some extent, and the oxidative burst can in fact be toxic, but it doesn't alter phagocytosis. And like I say, we're stretching me a little beyond my my understanding of immunology, but my understanding is growing by the day. I'm much happier with the histology here, because I've done a lot of histologic analysis. And so what you can see from these is that there's actually a reduction in the infiltration of the leukocytes without actually interfering with mucus production. And the mucus is really important for lung function, so that the cellular infiltrate is shown by the arrows. And the arrowheads show, although it's a bit of a stretch, but they're looking at this mucus cell production. So they make a good case. They publish this in scientific reports. That journal has very rigorous scientific review and peer review. So I think that, and by people who are indeed experts in the area. So I think that we can accept a lot of what they're saying here. This is the one that I just found, let's see, yep, just the other day. It just popped up on my sort of feed of what's new in COVID-19. And so what they saw is that, unlike a lot of other viruses, that COVID-19, there's this unique and inappropriate inflammatory response, so that there's a low level of type 1 and type 2 interferons with an elevated level of the chemokines and IL-6. So that what they're saying is that this reduced innate antiviral defenses, coupled with this exuberant inflammatory cytokine production, is part of the pathophysiology and the driving features of COVID-19. And this is the graphic abstract actually from that paper, basically showing essentially the same sort of thing, where you've got the healthy response. I'm sorry, I think you've got different, move myself over here. Yeah, your standard respiratory viruses and the response that you get in terms of inflammation and immune cell recruitment and a potent antiviral state with interferon beta and gamma. And in contrast, COVID-19 gives you this immune cell recruitment, but it lowers some of the interferons here. And so it sort of puts itself at a distinct advantage. And so that what they're saying here is that this, like I said, the response here is imbalanced with regard to controlling virus replication versus activation of the adaptive immune response. Because of this dynamic, treatments have less to do with the interferon response and more to do with controlling inflammation. And the data indicate that numerous chemokines and interleukins are elevated in COVID-19 patients. So they're saying that future efforts should essentially focus on drugs or treatments that have immunomodulating properties, which is essentially the focus of this entire symposium. What I want to say as a sort of caveat here is that we have to be careful, because this immune system regulation and dysregulation is very different with this virus, and the timing, we know that the timing of treatment and are critical determinants in photobiomodulation. So Dr. Lyons and I have actually shown that photobiomodulation has this potential to ramp up the immune response. Now, you don't want to do that if you're already having a cytokine storm, if administered at the wrong time in the disease process. So what we saw is that we treated some of her mice in this experimental allergic encephalomyelitis model of multiple sclerosis. We generally, the work she has published and has followed up, the treatment is after the onset of disease, as the animals are starting to develop the disease course, are starting to show manifestations of disease. If they are treated for, I think it's 7 days on, 5 days off, and 7 days on again, she can essentially flatten the severity of that disease. When we treated these guys early in the induction of this disease, we exacerbated disease. We made these mice very, very sick, very, very fast. The other example is one from a sort of personal story that went into science. It's actually an interesting reflection on how scientists sometimes experiment on themselves and then go find out what actually is really happening. I have a colleague at Indiana University, and we were doing some work on looking at developmental effects, looking at the effect of photobiomodulation to protect against environmental toxins in a chick embryo model. And it works so incredibly well that my colleague who has polycystic kidney disease became an early adopter. And she decided that if it did this much to reduce anything negative happening to these chicks, it had to be good all the way around. And so she immediately applied the light to her back, right over her kidney, and immediately popped 2 cysts and ended up in the hospital. So, you know, it definitely advanced that disease. Now, being an incredibly good scientist, when she had recovered from this, she turned around and collaborated with a colleague who works in a model of polycystic kidney disease called Meckel syndrome. And what they showed is that they can accelerate Meckel syndrome with near-infrared, actually it was far red light. And so these Meckel syndrome rats were treated with light therapy, and disease, you know, and the severity was evaluated. And what happened was the blood urea nitrogen went up 6-fold in the cystic rats, so the animals that have these polycysts, but double that again with the photobiomodulation. There were, and catalase, the anti-inflammatory enzymes, glutathione peroxidase, catalase were reduced in the cystic kidney, and superoxide dismutase was upregulated, and actually cytochrome oxidase was upregulated. So essentially the cystic kidney is saying, I need more energy, let's, you know, upregulate mitochondrial function. And Photobiomodulation actually increased catalase and increased cytochrome C oxidase. And they're actually and theorizing, and their data support it, that essentially this detrimental effect could be associated with this change in blood flow, and that this tissue compression. So essentially what they're saying is we activated the mitochondrial electron transport chain with light therapy. And when we did that, we stimulated cytochrome C oxidase activity. We really increased the kidney's need for oxygen, but at the same time, these cysts are compressing the vasculature, impairing the oxygen availability, and producing more free radicals. And so by basically stimulating tissue metabolism, they effectively made the disease course worse. And they show it here. Let's see if I have two. Nope, I just had the one slide. So the best pieces of this information are, one, take a look at the normal, go to the bottom here and look at blood urea nitrogen. So it's very high in the cystic kidney, and very low in, let's see, in the control, right? With the LED treatment, you really see a huge increase, way above the, double, essentially, the cystic kidney alone. And the same sorts of things are occurring, the things that are occurring in terms of cyst volume changes, and also changes in fluid, and definitely the mice, or the animals, were in fact sicker. So this is my sort of warning thing. This is my team. We do a lot. We have a lot of fun together, and we have this is the light-treated animals that I look at visual responses in. Elizabeth essentially sort of runs all the labs, and several of these other people are former or current graduate students. And these are the generous people who have funded this work over time. And I'm done.
Well, you leave me a lot of time, a lot of questions that I want to point out. And one thing is that I have kind of cut my teeth on kidney and light. And I have treated many single kidney people and children with, they didn't have polycystic kidney, but it's very close to my heart. And we found that by treating those kidneys, we increased the blood flow to the kidney and allowed their function. I've had transplant people and everything like that. So when you're looking at polycystic kidneys and things like that, and cysts, remember, sometimes this stuff is encapsulated because the body can't really deal with it at that point. They don't have the energy to heal. So, of course, if you're going to give it more energy, it's going to do its best to heal, which is not
couldn't do.
It's not necessarily a bad thing. You know, as they said, if you eat chow mein and get toe mein and throw up, are you sick or are you well? You know?
Right, exactly.
And what I want to point out is this is kind of that you're touching on a very good subject, but I think that to say that it was so you went to the hospital, so what? I mean, the body is healing or it's getting sick. You have symptoms, but because it's the same scenario of a diseased body that has to heal. So you don't go from sick to well. You have to unsick yourself.
Right.
Part of it is going to be the detox. I mean, if you kill, like we talked about, if you kill the virus, you still got to get rid of all the dead stuff. If you kill the cells, you still got to get rid of all the dead stuff. And if your system's not up to speed, guess what? That's going to be a symptom and exacerbation. But I have to say, you pointed out two cases of the kind of the only detrimental effects. Good thing there's not a whole lot of light therapy. So good for you.
Oh, yeah, no, and that's exactly what I wanted to do. I mean, we use it in our house for everything from, bruises, wound healing. I treat the cats. I will treat just about anything moving with light. I convince Jerry I am like the Johnny Appleseed of light therapy. or it's, it's my hammer and everything's a nail. I have talked more people into doing work in this area, or just, encourage them. Jerry Lyons and I must have talked for 2 or 3 years before she did her first experiment in her MS model. And interestingly, that is actually an amusing story too, because I convinced her to do it. She went ahead and did it. She was sort of grousing about the whole thing. She calls me up after she's analyzed the data and she said, well, Janis, I think it made things worse. So I guess I'll just go back to the other stuff. And so that was that was early in the evening. I get a call at like 1 a.m. I reversed the treatment groups. Not only did it make things better, it made things a lot better.
Well, I wanted to say that's you kind of were one of the first labs that were looking at pre-treatment. So I want to kind of make a caveat and go back over your caveat because, I mean, giving it too straight and then doing the stuff, or, they gave the shot for the mice model and then gave the light 2 times type of thing. Okay. But what I'm saying is, if we look at the disease, if we look at the host immunity or the host disease symptom expression over the course of the treatment, you could say, oh, if I put it, I could put it as a pre-treatment. I could put it in as an anti-inflammatory treatment in the middle. I'm sepsis. I'm almost dying. You can put the treatment in there. So, I mean, timing, as you pointed out, is very important, and I think we still have to look at that. Now, I want to ask you, how about the natural light? I mean, you missed vitamin D as a. Oh
yeah, no, and I won't. I'm a big believer in vitamin D, and in fact, there's a wonderful paper that came out called "The Vitamin D Mistake," which essentially said our guidelines for how much vitamin D we're supposed to take is enough to keep us from getting scurvy.
That's correct.
But not enough to really make us healthy. I take 5,000 units a day, and my vitamin D levels are still, just moderately normal. When I went in, I was deficient. Correct. We're all deficient.
Correct.
So yes, and if you look at, I mean, I've pretty much kept myself, in terms of my science, limited to red and far infrared. Right. I'm sorry, far red and infrared, near infrared. But I've read a lot, and the work that Kellogg did with, not only cornflakes, but you had light therapy. Right. All over. I think he set up a lot of his stuff in Australia, and there are still awards that you can get, Kellogg's awards that are based on the light therapy. Finson, the ancient Greeks, the longest lived cultures on earth, get a lot of sun. So I am a big believer in sunlight and natural light. I think there's a lot to be said. Mike Hamlin's written some stuff on UV. I don't know nearly enough about it to even want to venture into that.
But so the thing that you could talk about is your research was really, looking at the eye, and the eye perceives color and light and everything like that. So you look at it as an application. What if you were putting the light in the nose? Would you could see a lot different, activity being specific organ type, quote unquote, organs.
Oh yeah. Well, I mean, all the work that's being done by Lou Lim, in terms of, Alzheimer's. I mean, they have a trial going on. They've got a unit that sits over sites on the brain, but then they also have the intranasal unit. If you want to reach the brain, if you want to treat Parkinson's, stick a light up your nose.
I'm saying if you pre-treated the nose, would it have wouldn't it have a positive effect for the autoimmune effect right there versus I'm going to put it on my lungs when I'm already sick type of question?
Yeah, no, I agree. There's definitely, this has been, several people have shown this. Jonathan Stone did some work on this. and I really think it started actually even before that. There's a systemic effect. I mean, the first time someone at one of these meetings, Dyson, I can't think of her first name. She's a brilliant woman. Anyway, when Mary Dyson told me at one of these conferences that I first went to that it wasn't just, that there was definitely a systemic effect, that what was going on in the eyes, I probably could have started some of that by shining light on the butt. And I just laughed at her. And then Jonathan Stone did the experiment with Parkinson's, where he did exactly that. I call it the rat butt experiment. He called it indirect administration. But anyway, they wrapped the little guys' heads up in foil, and they had given them a drug that causes Parkinson's, MPTP, and they treated just the body. And then they treated the head, and then they compared, and they got about 60% of the effect that they got from treating the head, they got from treating the body. They don't see a synergistic effect. So you can treat the whole animal and get one effect. You can treat the head and get a little better effect. You can treat the butt and get a pretty good effect all by itself. So, and pre-treatment, a lot of the work that Christina Walter and her group has done at the Australian National University has been treating before. Ricardo Natali has actually looked at non-coding RNAs, some of these RNAs that are really part of transcription factors, and they go up if you pre-treat. So there's definitely, it's not like you're not having an effect. You're sort of gearing the system up. I mean, I'm hoping at some point in medicine we'll get to the point where instead of treating, I mean, we got it all backwards. We are a sickness treatment society. We are not a let's stay well group. And what we want to do is keep from getting sick, not treat the symptoms once we've got them.
Correct. So if you could use like I said, the full body immune boosting aspects we know exist.
Yep.
And the intranasal, blood activation and the things that we're talking about, viscosity changes and blood changes. Those are positive effects that I believe that they could be, staging it, like you said, the timing of it is important, but treating far away from let's say, you could treat a little septic lung if you're in the hospital and they're doing the research on that stuff now. But remember, we're treating the human, not the disease.
Right, exactly.
Virus. And I think that's kind of where you got to play your cards. Now, your quote, I wanted to quote you correctly. You said the entire world is answering one question. What was the question?
The question is, what the hell is this virus doing? I mean, and how do we stop it? This is the first time. Yeah, this is the first.
And I think that the group of scientists that have put their mind together on the light and photobiomodulation versus COVID-19 is doing an excellent job at bringing these things to light, bringing to light. That can change these systems. We are set up to change this thing, to look at our immunity changing. Because remember, it's the body that has to heal, right? It's our immune system that is going to have to, one way or the other, kick in to wipe this thing out, right?
Right, exactly. And it might be good to just say, hey, can we prime you so that you're ready when this sucker comes?
And then the other studies that are going on. Like when you talk about the lymphopenia, which hasn't really been touched on too much, don't we see lymphatic applications? Don't we see an increase in lymphatic activity in the lymphocytes and the leukocytes?
Yeah.
Photobiomodulation. I mean, there's a whole little thing on that would be a part of that research. Of course, the anti-inflammatory thing, but isn't there enough research to show?
I think there's some on that too. Again, like I say, we're stretching me at my scientific limits. So I'm learning as we go here. And
I think that we found that the blood treatment is the one that has the biggest activation on lymphocyte activity.
Right.
So,
And the Russians have been doing this forever. They have been threading little LED units, little lines into your blood, and actually I'm trying to think. I think there's a couple groups in someplace in Northern Europe that are doing the same sort of thing. So
Germany,
that basically, Germany. So you come in and you get this hemo photobiomodulation treatment, and so it gets and the guy that I started a lot of this work with, the guy who built the Warp 10 and the units that I worked with, Ron Ignatius of Quantum Devices. Ron used to put he used to grab his little Warp 10. I have one right here, and he used to put one like this and like this, and he would just zap the vascular system going to his brain. And Ron was a huge man. I don't know if you ever met him, but Ron was probably approaching 400 pounds. He was he was a tall guy, but he was much wider than he was tall. And it was a miracle that he was alive. And I think part of that miracle was that he treated himself with the light. But he would, it was, and I
agree. We have many different ways to apply it and many different ways to attack this. But we know that light is illuminating immunity.
It is. All right.
Thank you, Jan, for all your contributions. Thank you for your hard work. I want everybody to follow you and on your at least on your LinkedIn, because I'm learning a lot. You're doing a great job. Thank you.
Thank you. Thanks a lot. It's good talking to you.